So I'm part of Skip's group, but I'm branching off and beginning my own line of research. And so what I want to talk to you today is one of my focuses is on continuing to mine existing genomic data for new information and ways that we're doing that. And so, if you'll allow the analogy, I like to think that in the decade or so since genomics has emerged, we've gotten really good at getting what I call a first cold press genome, and that's like all this data exome sequencing. We've gotten the tools and the ability to really extract a lot of great information really fast from genomic data. But one of the overarching hypotheses of my goals is to keep pressing and see what else we can extract after kind of the first extraction is completed. And so one of the ways we're doing that is by looking through what goes into the unmapped reads from genomic and exomic data, and saying, can we start piecing together additional information from what's thrown into the trash heap, thrown into this unmapped reads basket. And so there's a lot of different reasons that reads are thrown away into the unmapped read bin, if you will. And a lot of that is low quality from sequencers, but sequencers are getting better and better, and there's actually being high quality reads being discarded into unmapped read no man's land. And really we're being able to then go through this and extract these and piece them together and find that, yes, these are human sequence, we can get them in high quality and start rescuing these things, and we hope to eventually start getting this at enough depth to integrate these back into the human reference and start adding these additional sequences to the human genome. And you'll see that actually, if you look here, when we've done this, and we're focusing on microsatellites, because that's the tools that we've developed right now, and there's a lot of information here. But if you look at the microsatellites that we've looked at in these unmapped reads, we can see that if you look at the red bars, these are the ones that are kind of known in the current reference genome. And when we look at the unmapped read category, we start seeing that there's a lot of 5-mer microsatellites that are just being heaped into this unmapped reads. And so we're starting to be able to analyze this and say, what are all of these? What purpose do they serve? And we think that many of these are centromeric microsatellites, and we can start piecing together these. And so I want to touch real quickly on also another way we're doing it, and it's been recently published, so there's more information out there on a new measure of somatic microsatellite variation, where we're looking beyond the genotype to look at additional alleles that may be present in sequencing data to see if these additional alleles can give us an idea of the amount of variation within an individual's genome. And we're finding that people with known defects in DNA repair mechanisms have a different pattern of somatic microsatellite variation than do healthy cells. And so these two ways, we're kind of extracting more information from genomic data.